Hello and welcome to this lesson for higher biology, which this time around is on genomic sequencing. So previous to now we have looked at the processes of evolution and how a new species is formed. In this lesson we're looking at genetic sequencing and how that's contributed to our understanding of evolution. And next time we'll be looking at personal genomics, how this can apply to individuals. So what we're mainly looking at today is understanding how the sequencing of organisms' genomes has led to our understanding of evolution. And what you're required to do is explain what genomic sequencing is and how it's used to compare different species' genomes. You need to be able to state the meaning of both the terms phylogenetics and molecular clocks, and you need to be able to list the main sequences in the evolution of life and give the two types of evidence which have been used to establish this. The process of genomic sequencing is what we do when we're trying to sequence the entire nucleotide sequence of a whole gene or an entire genome. And what we mean by nucleotide sequence is literally just the order of the bases, the specific A's, G's, C's and T's, the order of those in each gene or possibly in the entire genome. And this predominantly uses computer programs which are used to identify different sequences, and then they work out where they overlap in order to put together one full sequence. And this genomic sequencing has been carried out for a number of different organisms as well as for humans. So some organisms which have been studied have very similar genetic sequences to humans, and as such have been used in laboratory studies. And the main ones that we consider are things like E. coli, using mice, and using fruit flies tend to be quite common model organisms that are used. In order to do this, we use both computer and statistical analysis, and that's the field of bioinformatics. And what that's doing is comparing the data from different species. So if you were to look at this one, having sequenced the entire human chromosome, what they've then been able to do is look at the mouse chromosome and seen where different sections match up. So if we're looking at, for example, mouse chromosome number 6 here has got a big red stripe, and that's come from part of human chromosome number 7. The rest of human chromosome number 7 is over here on mouse chromosome number 5, and so on. And the uncoloured in sections are sections which are unique just to the mouse. They're not necessarily present on the human genome. So that shows you just how much similarity there is between one species and another. And this is done, as I say, through using the computer programs and certain statistical analysis programs, and that's what we refer to as bioinformatics. And when they've compared all of these genomes, that has revealed that the DNA sequences of quite a lot of important genes are highly similar, so they're conserved from one organism to the next. For example, these might be genes which are coding for proteins involved in respiration, or they might be particular enzymes that are involved in most species. And these similarities have meant that scientists tend to focus on sequencing the genome of relevant organisms, so pest species, for example, or research species. So if we were to look at the conservation of different genome sections between a human, a mouse, a chicken, or a zebrafish, if we were to look at our human, it's got 2,963 unique genes, it's sharing 1,602 with just the mouse, 892 with a mouse and a chicken, 10,660 shared across all 4 species, and so on. So when we compare all of these different genetic sequences, that allows us to demonstrate how related different groups of organisms are. And that leads us to the field of phylogenetics, and that's where scientists look at the evolutionary relatedness of different species. Often this is involved in analysis of ribosomal RNA, and that allows the nucleotide sequences of those to be studied because they're shared by all living things. And comparison of those sequences has given us clues to the evolution of all living things, and has demonstrated that all life belongs to one of 3 domains. So that's the bacteria, the archaea, and the eukaryotes. And if you remember from before, eukaryotes are the types of organisms which have cells with a nucleus present, whereas bacteria and archaea, usually prokaryotes, don't have uh, nucleus. This can lead us to develop something called a phylogenetic tree, where what we're looking at is a group of closely related living things accumulating different mutations, so spreading out into different types of species. These nucleotide substitutions are gradually altering the genome, and what we can then do is look at the number of differences per unit length of DNA, gives us the difference between two genomes and allows us to get a measure of how related two individual genomes are. So if we were to look at, for example, these ones are quite closely related, they're quite close together on the tree, so although they've had a lot of mutations from their original one, over the units of time, the amount of time they've had to consider these mutations, they've stayed fairly similar. They've only got a few mutations separating them. Compare that to with these guys and the tyrannosaurs over here. Much further apart on the tree, much further branches, so many more mutations over unit time, making them very different. So that would look something like that on a much more simplified diagram. So we've got our original common ancestor, every 1 unit of genetic change. So there's been one change there and another change there, and that's separated species A from species D. Then further up the tree, we've had another mutation along here and another one here, and again that's further separated species A and species C. And then along here we've had one mutation going this way, one mutation going that way, separating out species B and species C. So the closer together specific species are means they're more closely related, and the further apart they are, such as A and D here, they are less closely related, so they've got very different DNA sequences to each other compared to ones which have got far more similar DNA sequences to each other. One of the things this allows us to look at is the fact that mutations can accumulate at a steady rate over time. Now, as we've said before, mutations occur randomly and rarely, but the rate of mutation tends to be fairly constant. Therefore, the number of nucleotide substitutions that a genome accumulates, so going from our original gene to our different mutations along here, is regarded as being proportional to the amount of time. So if this one has had 3 mutations, that gives us a certain amount of time must have passed to get from there to there, for example. So comparing this data to fossil records gives us what we call a molecular clock. That gives us information about how long ago the most common ancestor of the species existed, and also tells you the sort of sequence in which the different species evolved. So for example, we're able to see here that species B and species C share both of these two mutations. So that tells us that this mutation and this mutation must be our most recent one. Comparing these genomes with species A, they don't share any of those ones, so that suggests that these two mutations and these two mutations occurred at different times and occurred in different species. So these must have occurred to the separate species rather than here where this one at the top, the yellow one and the blue one, have occurred to this species. So that allows us to figure out which ones came first and which species have been originally related to each other. Worth pointing out actually that the greyed out sections, so here and here, are actually areas where they would match up. So all of these species have got identical genomes. The highlighted and coloured sections are the new mutations which have occurred. So using these molecular clocks and using our fossil records, we've been able to establish several different key events which have occurred during the evolution of life on Earth. And there's a rough timeline there to how many billions of years ago, and some of the key highlights of when different organisms have evolved or different areas of organisms have evolved. Now, you don't need to know specifically how many millions of years ago all of these things happened, but a rough idea of the sequence is worth it. So, beginning with we have life, then we have cells similar to prokaryotes, we've got the last universal ancestor, then we've got prokaryotes which can photosynthesize, eukaryotes, multicellular organisms, animals, vertebrates, and then land plants. as our last one. So those are sort of key points in that evolution timeline and the ones that you should be able to try and remember. As I say, you don't necessarily need to link them to how many millions of years ago that was, but remembering the key points that have occurred. So you should be able to explain that genomic sequencing is when we take the sequence of the entire gene or genome of an organism and figure out what the individual nucleotides are and what order they go. You should be able to say that phylogenetics is when we are looking at the evolutionary relatedness of different species, looking at how the mutations have occurred over time. That molecular clocks are used in order to consider the number of nucleotide substitutions that have been accumulating in a genome over a certain amount of time, and that we've got several sequences in the evolution of life, beginning with the evolution of life and finishing off with the evolution of land plants, and that this has been established by the use of both molecular clocks and fossil records. As ever, feel free to ask any questions as we go over this in class.